So we're going to videotape this, and I'm going to try and take advantage of this feature where I can pause it, so we don't just have like 5 minutes of you guys thinking through it. So remind me to do that. Come back to this and do that. All right. So look, what we are doing is now looking at criteria for epithelia. There's really 6 that we want to see. All right. So now it says 8 subcategories. I'll elaborate on that a little later when we get to endocrine and exocrine. All right. So I've said a couple times, you want to, for each of these, you want to be able to get the name, obviously, its location, all the locations, and its function. All right, and then what you need to do is view a cell from different angles, and we'll show you. And now on Blackboard, I got a whole bunch of practice slides, probably 23 epithelial slides for you to look through, and some of them are also going to be in these little packets, and then we'll review a little more before we do our quiz. So when I look at this, I think about there's two things that we're kind of doing here. And this is the easy part, and I've said it a number of times. I don't want to be a dead horse here, but if you're trying to discriminate between one versus the other, this is shape, and then this is number of layers. Okay? So, hey, apical surface, this up here, is that going to be closer to what? What's it going to touch? The outside, the free surface. That says apical. Associate with apical, free. Associate with basement membrane is deeper, or it's going to be more. You're going to associate that. It's going to connect. So think about your CT, your connective tissue. All right, and then the other thing is layers. So simple is 1. This is greater than 1. That's supposed to be a greater than sign. Greater one layer. So this, I'm not going to spend as much time on this. I'm going to go right into kind of what I said you needed to know. Be able to identify it, draw it, labels. But here's your simple squamous right here. Or here's your simple cuboidal. Here's your simple columnar. And then you got a little indentations here. We'll talk about that shortly. And then this is the generic. But then I'm going to give you some practical examples where you see, like the transitional is one of the complex ones, where the shape is different depends on the location, and whether you're talking about fluid pushing on it or not. Full bladder versus empty bladder, the shape of the cells change, and that's a necessary thing to be able to have them to be able to expand and stretch. Or if they didn't, then you'd have a bunch of people with urinary infections or dying from an infection because their bladder exploded. You know, everyone jokes about it exploding. No, it actually doesn't explode. If you're at capacity, then involuntary things happen, like something I'm not going to elaborate on. Okay, so this is my point here. If you want to be ready for any type of evaluation, you ought to be able to look at a picture, see the shapes, be able to, find the nucleus because they're going to be stained, but then see the little shape, like a flat sheet of cells, okay, which is what is unique. But then we're going to take this and flip it on its side and look at different views. Look at this is the peritoneum here. This is the abdominal cavity, and you'll see the lining there. Well, we'll get different views. All right, simple cuboidal. Hey, what is this open space called? Huh? Lumen. And I tell you, people, you always have stuff in anatomy where like there's a whole slew of stuff you got to know. There's that one you remember, and it's like you see the explanation point where people are like epithelia, stratified, squamous, lumen, and they get all excited when they say lumen. So, all right. So this is a guide. We're gonna run through this simple columnar. Here's some locations, functions. All right. And what I want to do is go through the next slides, and we'll add some of this stuff. And some of it is right from here. There's some other aspects that you may have not seen. So, stratified squamous. Look at the locations. Now, this one I won't rewrite all of them, but from the lining, the covering of the mouth. Why do you want to have this several layers? Because you want to have those several layers because that's the protection. You got abrasions going on. You're scraping and cutting. You swallow food. How many have felt that when you were like in the lining of your esophagus when you're like maybe eating popcorn or Doritos or something, constantly, but you're not drinking water and start scratching the back of your throat? There's a good reason. You're sloughing off cells, but you're not going to die from it. Right? So there's structure and function. This is a basic principle of any biology class, which this is. This is a biology level 2 class, right? Structure and function. So, and then now we're adding in location. Okay, so any other general stuff here? This is what I was talking about. See the difference with transitional here versus here, right? Empty versus full. Empty bladder versus full bladder. This allows for stretching here, right? And expansion, structure and function. Okay. And then the pseudostratified columnar. So what we're going to do is go through some of these now, and so we're going to run back through them, and I'm just give you pointers from my notes, and I'm going to draw some stuff in here. So again, structure and function. So look, I've given you the title part here. Doesn't look like the video was getting that part. So, all right. So let's look at this. What can you label here? So this is like an air pocket. Air sacs along. What do we call that? Anyone remember? Well, bronchiole is a lot larger. The actual sac is the alveoli. Right? And again, that's not on the quiz, but it's also, as we start throwing in this stuff, we'll know that. Right? So right here, alveoli. Okay, so this is a cartoon. It's a schematic of it. This is an actual photomicrograph. Right? And remember, one of the categories we said about epithelia, those 6 things that we listed before, is that they're tightly bound in sheets. So you take the sheet and you use it to wrap around things. All right, so let's draw a little something here. So here's all right. We say they're flat cells, but that doesn't mean they can't bend. And that's really what you're seeing here. So this is a squamous cell. Each one of these is the things. Each one of those little dark dots is the nuclei. So take this, shrink it down, and each one of this is squamous epithelial cells. Simple squamous epithelial cells. Okay?
Wait, is that the alveoli that labeled AR?
Oh, well, no. This is alveoli. This is actually air here. There's air here, and then now what's going on in between each one of these cells? So imagine now I would connect another one. All right, let's connect. All right. Does this make sense? All right. So this would be your air, and then what's also, or this could be, if you're, we're talking about simple squamous. This could be a blood vessel too. There could be blood in here. And how do you get things in here? Let's see. Things are going to diffuse in and out. Structure and function. So for things when you got this flat surface, they're going to be able to diffuse through rapidly, which is important. You want your air and your oxygen in, carbon dioxide out. You want that ratio to go at pretty steady state and a rapid state. Okay. So simple cuboidal. Again, I'm just going to give you a couple facts about each one of these. All right. So think about simple cuboidal. These ones, like they're going to form tubes. All right. So you take a cube. If you were to take a flat sheet and put a little small cube like this. All right. Actually, that's so there's your flat sheet. Just ignore yourself. All right, and then, so this is the cuboid cell. And I don't know why it's not picking that up. And then, I take another one and put another one and put another one and put it all the way through. So imagine this being covered up with, okay. So this is like the basement membrane. This is the basement membrane. This is the cell. Now I line up a whole bunch of these, and then I fold it into my tube. All right, does that make sense? So cuboidal cells do a really good job with this. And here's some examples of where you're going to see them. So imagine the kidney tubules. But this is they're small. Tubules are small. And if you're talking about this specific function, there's three things. It's got to be able to do this. Help collect urine. Not produce it, but collect it. All right? And sometimes store it there until it's ready to get rid of it. And prevent leakage. Prevent it from leaving. It's going to prevent it from leaving. All right? And you'll see this in the tubules here, and you'll see that actually in the ureter. This is where you'll see this structure here. All right, that's it. This is a ureter. This is the bladder, urinary bladder, and then this should be the urethra. So, all right. So think about simple columnar. The easy part for you guys is going to say, oh, that's columnar, that's stratified, squamous, but then start adding in, hey, here's its function. And then what I'm telling you is if you start thinking about its shape, structure and function are tied in together. All right, so you guys know about microvilli? You can't always see it here, but this border here, so inside here, that's the lumen, that's where the food is going to be. These cells are going to be part of the process of them diffusing in. And now down here, so like, right, we're going to follow, like in this area, somewhere you're going to have a blood supply, all right? Or blood vessels, I should say blood vessels, not that abbreviation, all right? I am according to that. All right, so we'll say you're going to have your capillaries, your veins, because we're talking about getting it to the rest of the part, back to the heart, and then to the rest of the body. So look at it. These cells actually have microvilli, so they're cytoplasmic. So this is the microvilli right there. It's a cytoplasmic projection of the cell membrane. All right? So again, think about the whole purpose. These aid with digestion, in particular, absorption, and there's some secretion that goes on as well. So you're thinking protection when you're thinking absorption, and the defining factor for absorption is these microvilli. All right? And then there are some secretion. There is some secretion. All right, don't confuse microvilli with cilia. That's it. The main thing is the absorption, but it is protection and secretion. All right, so these are hair-like structures. Now, one of the things you see with pseudo-ciliated columnar, all right? Remember, E stands for epithelium. Okay, so here's your cilia. They also are right next to. Hey, if this is a cilia, and you also have this structure called a goblet cell, is this going to be mucosa, serosa, cutaneous? Look at the location. Mucosa. All right, you have specialized cells called goblet cells. They produce mucus. Now, look, this is actually a protection mechanism. This cilia, all right, that lines the trachea, Right, so this is where where you're going to find all of this. Right, this is going to protect you against the outside, because why? The outside, that's an access point for the outside. So this is your first line of defense for things like dust, bacteria, virus, and also some other microbes that aren't bacteria and viruses, like some protists or mold or fungi. Right? So we can even say fungus, or we can even say protists. For that matter, we can even say tiny microscopic animals. All right. All right. So look, you got these structures they're sweeping in, and one of the reasons why they're kind of doing this. All right. They're moving back and forth. So I remember explaining this to a student a couple years ago about why is it bad to smoke, or why is it a smoker who has so think about cilia. If I'm a pseudo-stratified columnar, all right. So I got my cilia. This is the cilia dance, by the way. All right. So imagine my limbs being the cilia. So I'm in the respiratory tract. I'm there, and these little particles say this is something. Right? This is something bad, so I'm sweating, I'm like this, and we switch it so the person, and they get rid of it. Alright? So the smoker, what the tar does is it retards this movement. So, hey, no. Yeah, I should probably have one of you man the camera so I can.
No one wants that.
Well, yeah, that would have been a good idea. I'll remember that when I travel into the past. Okay, so. All right, so I'm the cilia, but then the smoker, instead, you're a non-smoker. Smoker? It's real tricky. No, I shouldn't go there. All right, so now, but the problem is, everyone has to sleep, right? So they go to sleep, and then the tar's gotten out of their body, so they wake up, and what are they doing? These are like, they're making up for all that time, and they were like, so they're like blue dance. They wake up. If you've been around a smoker, they feel like they're coughing up a lung. Well, there is a little truth to that. They're coughing up mucus, and their cilia are finally acting and getting rid of that stuff, passing it around to. It's easier to get it out here than swallow it, get into your alimentary canal, and send it out there. Those are the choices. All right, not to be too crass, but biology is wonderful. Okay, so if this was last year, we'd already be done, but we still have 40 minutes. Okay, so I'm not going to talk a lot about this one. This is stratified squamous protection. I kind of talked about your esophagus, so think about this is the main thing for this is protection. All right, so let's jump down to this one. Transitional. And how do you well, I got to mention something about stratified squamous. You use the determining factor of whether it's flat versus this. If you look down here, don't these basement cells, they don't look flat, do they? So there's the basement it's already there. There's the basement membrane. What is this then? All right above this would be the apical surface. All right, so that's like up there. That's the apical surface. We as anatomy, we use the flat surface. We use this area as your determining factor of whether identifying the shape of the cell. All right, I'm kind of dancing around saying that. Remember, you've got your basic three shapes. Well, if you really studied this, you would see as you go further down here, they actually look more cuboidal or like a squished columnar. It's because they actually are squished because of the many layers. All right, so you'll use the top layer to determine if it's stratified. And really, here's a good example here. See over here, they look like they're flat. They're more bulbous or circular, but then down up here they're really smashed. And here's one of the things I like to point out about stratified squamous is you have this is where you have the fastest mitotic rate. You have a very high mitosis is going really fast, right? So the mitosis rate is very high here, but up here, these cells are actually dead. then so the outer cells, outer layer cells. When we get to the integumentary system, we have a breakdown for all of this. Right? As we get down into connective tissue here, we get into the epidermis, and then this will be the dermis, and below that will be the hypodermis. Okay, so I already alluded to this earlier. There's really not much to say other than the difference with here is you have these what we call these cells are kind of bulbous, right? But they can change shape when it's expanded or when it's expanding because it's full of urine, or it's gotten rid of the urine. So that's pretty much all I wanted to mention about that. I do want you to know you ought to be able to recognize this to a location, right? Most important one is the urinary bladder, and the tubules that are part of the ureters, right? Because they can expand. Right. So, all right. So that's. We're going to go ahead and kind of stop here.